hope all of you had a heavy lunch. Was it good?
Yes.
No? Okay. First of all, okay, anyway, Dr. Abhishankar is gone. I think you would have already got a brief introduction about me. I am a faculty in Department of Chemical Engineering. I joined a year back at IIT Tirupati. Before this, I was doing my postdoc in Stanford University. I did my postdoc there for 4 years. And before that, I was doing my PhD in IIT Roorkee. And then I did my MTech there itself. And before that, I did my BTech in biotechnology from Kumar College, which is down south in India. And okay, I think that should give you a brief idea. But my bachelor's is in biotechnology. Then after that, I did my master's in nanotechnology. And my PhD was again combination of both. I could call it nanobiotechnology, probably. And then a postdoc again, pretty much on the similar lines, like nanobiotechnology, combination of both. So if you are expecting that you will see a lot of chemical engineering aspects in this presentation, I'm sorry to disappoint you. There will be a lot of life science and biology stuff in this presentation. So yeah, let's get started. So, so before I start my lecture, I just want to know the composition of the audience. How many of you have a background in life science or biology? Anyone?
No.
Except you?
Biomedical.
Okay, biomedical. Okay. Okay, based on that, I have to elaborate on few concepts while I go through the okay, that gives me an idea that the majority of the audience is not in life science background. So yeah, we'll go through accordingly. So the topics for today's lecture, I think already for the past 3 days, you would have had an excellent course of different lectures from different experts who would have probably spoken on 3D printing or biodegradable polymers and so on. But in the next 1 hour or so, I'll keep it short. So I'll say 1 to 1.5 hours or so. I'll be talking about tissue engineering and scaffolds for tissue engineering, and what are the key aspects of this scaffold that determines their success in their application for tissue engineering. Okay, so to get started, first of all, I would like the audience to understand why do we want to learn tissue engineering? What is the role of tissue engineering and what is its importance? So in order to understand that, I would like all of you to imagine a hypothetical situation. God forbid, this is a hypothetical situation. So the situation is such that, imagine that one of your near or dear ones is sick and you're taking them to the hospital, and as soon as you reach the hospital, a lot of tests are done, blood test, urine test, all the steps are done. And then at the end of all the series of tests, physician collects the results, goes through the results. Then this physician comes to you and mentions that this patient is having a heart failure. Okay. And when he tells you that, he also tells you that the only solution for this patient to survive is that he should get a heart transplant. Okay. So it doesn't end there. The misery goes beyond that. When he tells you that it will take more than a year for a patient to get a suitable heart donor. Okay. And the patient doesn't have 1 year to undergo this transplantation. So this adverse situation happens throughout the world every day. And an crucial example of this situation is what is indicated in this plot. What you see over here in the y-axis is the number of people, and what you see in the x-axis is the different years. And as you can see over here, in the year 2019 itself, there are around 110,000 people in US alone who are waiting for suitable organ donors. And at that point, if you see the organ donors available, they are just 20,000. So there is a huge gap between the requirement of organs and the person and number of people who are willing to donate their organs. And as you can see, the more frightening aspect of this plot is that, as you can see, this gap continues to widen with every year. Okay, so we can expect the impact to be much higher as we move forward from 2024 to 2025 and every year. And the gap is expected to widen further and further, which means there will be less organ donors and there will be a lot of patients who would need a new organs. Okay, so let's imagine the same hospital situation in future. Let's say 5 or 10 years from now. Same thing happens. The doctor, the physician after going through a lot of tests, the patient collects the results and then he goes through that and then he comes to you and tells you that, okay, again, the patient is having heart failure and he needs a new heart. But this time the case is slightly different. What he tells you is that, don't worry, this is not a big deal, I'll take care of it. That's pretty weird, right? How can you expect a doctor to be so cool and confident when he has a patient who is having a heart failure and there's no suitable organ donor? There is a reason behind his confidence. And you get curious and you ask the physician, how come you are so confident and cool about it? He gives you an answer saying that what he has done is he has collected some cells from the patients and he plans to grow a fully functional heart in his lab within a time span of 2 to 3 weeks. Okay, and this beauty can happen only with the help of tissue engineering. Okay, and what he does is within 2 or 3 weeks, the fully functional heart will be ready in the lab, and maybe the patient can come back in the 4th week, do the heart transplant, and get back to his usual lifestyle. Okay, so simple. And this simplicity can be only rendered by the field of tissue engineering, where you can collect the cells from the patient and generate a complete functional organ in the lab, and then implant this functional organ back into the patient, so that there's no need for an organ donor. Okay, so
any organ can be developed, sir?
Yes, we'll talk about few organs which have been developed in the lab, case by case study of different organs, and we'll also talk about the success levels of each of these approaches. Yeah, we'll talk about each of those organs in the subsequent slides. So the ultimate point is, tissue engineering is the big change we can expect, which can overcome this gap. Okay, so I would say the change has already begun, and every change is started by a breakthrough invention or a discovery. Okay, and for the field of tissue engineering, this discovery or the breakthrough was achieved by this mouse called Vacanti mouse. Has anyone of you seen this mouse? Right, it's pretty famous image. In fact, I would say there was a separate photography session organized with the press to capture this mouse. What is so special about this mouse? This mouse is called Vacanti mouse. What is so special about this mouse? What could you see in this image? Ear. What you have on the surface of this mouse is a human ear which is growing, and the mice is also living. Okay, so this mice was engineered by a scientist called Joseph Vacanti. Okay, Joseph Vacanti, and he is a researcher in University of Massachusetts, and he did this in the year 1996. What he did was, he prepared a scaffold which is made up of 4 polymers, that is polyglycolic acid and polylactic acid. And this scaffold was fabricated such that it mimics the exact topography of a human ear. Okay, and this copolymer scaffold was porous in nature. And what he did after fabrication of the scaffold is that he seeded around 1.5 into 10 power 8 chondrocyte cells. Chondrocytes are the cells which give rise to cartilages. Your ears mostly doesn't have any bones. It's all made up of cartilages. Okay, so these chondrocytes are the cells that cartilages are composed of. So he collected 1.5 to 10 power 8 cartilage cells from the knee of a cow. Okay, he collected these cartilages from cow's knee, and after collecting these cartilages, he implanted or seeded these cells on top of the scaffold which he had fabricated. Okay, upon seeding, he allowed the cells to adhere onto the scaffold for a duration of 4 hours or so in a CO2 incubator. And then after a while, like around 12 hours after seeding, what he did is he transferred this whole construct inside an nude mice. What is a nude mice? Anyone who has an idea what's a nude mice? Okay, nude mice, usually every organism has its own immune system. So as soon as you introduce anything new into that organism, its body tries to reject that. Okay, that's the major reason for most of the organ transplantation failures. That the host patient who receives the new organ, his immune system recognizes this new organ as a foreign body. Then all the immune systems tries to attacks that implant and then eliminates it. So that is the main reason for the failure for most of the organ transplantations. So in order to avoid that scenario, what we have is an engineered mice, which is called nude mice, not just because it doesn't have a hair. It's called nude mice because it doesn't have any sort of immune cells, no B cells or T cells or no immune cells. So you can basically introduce anything into the body of this mice and it will not be rejected. Okay, so ultimately this scaffold, the copolymer scaffold seeded with this chondrocytes, was implanted subcutaneously. Subcutaneously means underneath the skin. You can see that it's not the ear implant, it's not within the body of the mice, it's just beneath the skin. And then they sutured it. Okay, and this mice continued to live for around 10 to 15 days. And what they did after this duration of 10 to 15 days is that they euthanized the mice, obviously, and then harvested the ear from this mice, and then studied what happened to these cells which were seated on this scaffold. So what they saw is that these chondrocytes which were seated on the top of the scaffold were able to grow throughout the scaffold, and in fact they could see chondrocyte nodules just like our earlobes. Okay, so it was a pretty interesting start where this was a first attempt when someone tried to develop an organ in the lab. But in this case, they could develop an ear, but the drawback is this is not a functional ear. This is just for aesthetic sense. It gives you a typical texture of a tissue or a skin, but this ear doesn't function like a typical ear. You cannot hear the sound with this ear. It doesn't have the inner components of the ear, the eardrums and all those things. But obviously, it can be used for an aesthetic sense. Okay, in case there's an accident and someone loses the ear, I think he would be happy with having an aesthetic ear, because he'll have other ear to hear it, but at least an aesthetic ear would do the purpose for him. So in those cases, this would be an advantage. But this was a very good start, I would say, because this was the first attempt where people tried to grow the organs within the lab and then use them for plastic surgeries and all. Okay, so now that we have gone through the idea of how this vacante mouse worked out, we have a rough idea what happens in tissue engineering. You get the cells, seed them on the scaffold, let the cells proliferate on the scaffold, and once the cells are proliferated, you implant into an organism or in any other living animal. So let's understand what is tissue engineering. So tissue engineering, what is the definition of tissue engineering and when it started? So it all began in the year 1996 when two of the scientists, that is Joseph Vacante and Robert Langer, published an co-authored article in the journal Science, and they defined what is tissue engineering in this article. And they call tissue engineering as an interdisciplinary field that applies the principles of engineering as well as life science for the development of biological substitutes. And these biological substitutes can either restore, maintain, or improve the tissue functions. So this is the definition which they give in this science article, which was published in the year 1996. So the interesting part, which I want to highlight in this definition, is the term interdisciplinary. In fact, the two authors who came together to define this tissue engineering term, that is Joseph Vacante as well as Robert Langer, stand a true testimony to the word interdisciplinary. Joseph Vacante was a clinical physician, and Robert Langer was a chemical engineer. Okay, so both of them, coming from a totally different research background, come together to define an interdisciplinary field, that is tissue engineering. And that is why usually tissue engineering requires a specialist from materials background as well as a specialist from life science background. If you miss out either one of them, there's a good chance that you'll end up with a failure product. Okay, and exactly that is the reason why this field is called interdisciplinary. And just like the initial authors of this article, each one had a different research room, completely different research room. He was working in a hospital where he was a clinician, Joseph Vacante. And Robert Langer is a well-known, very famous chemical engineer. His work was more with engineering scaffolds or coming up with new polymers that can be used for different implants. Okay, so both of them had to come together to make this Vacante mouse happen, and this was the first article which they published. But ultimately, this tissue engineering has different applications. So obviously, at this point, now you would understand that one of the most important applications of tissue engineering is that you can develop organs in the lab. Okay, and when you do that, you can overcome the gap in number of donors and number of patients who require a new organ. Okay, because you are able to develop new organs in the lab. Apart from these applications, there are some interesting applications which this tissue engineering can render. For instance, if a pharmacy company is planning to develop a drug, it has to go through multiple rounds of clinical trials, which involves a lot of animals, a lot of time. And because it involves a lot of animals, it needs sophisticated facilities and imaging stations and all those things. So everything gets costly when you have animals in the clinical trials. So if you're able to generate an organ in the lab, you can use such a system to test your drugs, and that will be cheaper, as well as the advantage is that it will not take much of time to do the testing process as compared to the case when you're using animals for the clinical trials. Okay, so these two advantages would ultimately end up in reducing the cost of the drugs in the market. Okay, so that is other application of tissue engineering. Apart from that, one other interesting application of tissue engineering which I want to show to you is this. It can be briefly summarized as from pig to plate. Okay. In fact, I think there are already some food products who have adopted this strategy. I just elaborate on the schematic, whatever is shown over here. What you see over here is a pig, and ultimately our goal is to create, what is it called? Pork, huh? What is it called? Pork, right? Yeah, okay, sorry. So ultimately, the purpose is to use, our point is to use tissue engineering to create an edible pork out of a small tissue extracted from the pig. Okay, what you have over here is a pig, and what we do in the first step is we collect some tissue, muscle tissue especially, from the pigs. That is called biopsy. Biopsy means you are taking out some specific tissues from your body. You collect some muscle tissues from the pig, and then grow it in the lab with growth culture medium. This medium would have all the growth factors or cytokines that is required for the cells to proliferate. Okay, so as the cells proliferate, it grows in size, and ultimately you will end up in a huge chunk of cell mass or a tissue. Okay. Now, since you are trying to grow the muscle cells would typically not be having the same characteristics if they are grown in a static condition in the cell culture lab. Okay. So you need to provide some mechanical stress to this tissue, and that is what is indicated over here. You have a system to subject this tissue mass to some mechanical stress, like stretch actions, so that the muscle cells look similar to the muscle cells of the pig as such. And then this can be converted into a pork. I mean, usually it is processed with certain additives or preservatives so as to make it edible for the common humans. Okay, so in fact, we have a vegetarian egg. I don't know how many of you have seen this product. There's a product in the market which calls it as vegetarian egg. And they have created that vegetarian egg using a similar sort of tissue engineering strategy. They don't kill any animal or they don't collect the egg as such, but they grow the cells which are there in the egg in the lab, and then they make the product. Okay. So literally the advantage of this strategy is that you don't have to kill the pig to get the pork in your house. Okay. If you have a tissue engineering lab, you can collect the cells, expand it, grow it into tissue, and then have it as a food. Okay, there's no harm in, you don't have to kill any livestock for that. Okay. So with that, I think now you would have roughly had an idea of cycle of events that happen in tissue engineering. The first step, suppose let's say if you're a tissue engineering scientist, a patient comes into your room for a clinic asking you that he has a damaged tissue and asks you to use tissue engineering to replace that. So the first step what you would do is you will collect some cells from the patients. Okay, suitable cells based on if it is a skin damage, you have to collect the skin cells, which is basically fibroblast cells. Okay, so you collect these suitable cells and then grow it in the lab, basically expand it in number. Once you expand it in number, then you see the cells on top of a scaffold. Now, scaffold is the main topic for discussion today. Scaffold is basically just a support material. It can be a polymeric material or a metal material. These materials basically provide the support for the cells seated on them. And why this support is required is that most of the mammalian cells, any cell which we have in our body, except the blood cells, they are anchorage dependent cells. If you culture, let's say if you take out your skin cells and culture in a bioreactor in a dynamic condition, which means let's say the fibroblasts are suspended in the growth medium and you're trying to grow that in a bioreactor, it would not grow. It will grow only when these fibroblast cells have a surface to attach. Only when these cells attach to a surface, they'll be able to proliferate or migrate or grow. Okay, so that is why these cells are called anchorage dependent cells. If you don't provide an surface for anchorage of these cells would not proliferate or grow. Okay, so most of the mammalian cells, except the blood cells, are anchorage dependent cells. And that is why you need a scaffold which provides a support for these cells to adhere and then proliferate and then migrate further. And that is what you do in this step, where you introduce the expanded cells on top of the scaffold. And apart from that, what you do is you provide additional growth factors or cytokines. So these growth factors and cytokines are usually used to mimic the in vivo conditions. In vivo conditions, in the sense, anybody who knows what is in vivo condition? No? Okay. In vivo condition is the condition which exists inside our body. These fibroblast cells are there in our body, right? But they are subjected to different conditions like flow of blood, exchange of oxygen, and there are different growth factors that are coming through the blood and reaching these cells. But all those things you have to mimic in the lab conditions, right? And that is called in vitro conditions. Any setup which you utilize to grow cells in a lab is called in vitro conditions. In vivo condition is the state in which you manipulate things within the body. That is called in vivo. So you are basically trying to collect the cells from the body and trying to culture in the lab. So this is in vitro, and these scaffolds are seeded with these cells. And along with the scaffolds, what else do we need? We need growth factors and cytokines. These growth factors will promote the proliferation and expansion of the cells. And once you expand a sufficient number of cells on top of this scaffold, you will have an equivalent tissue that can be replaced with the site of damage in the patient. So this, if suppose if you have a skin burn and you collect the fibroblast cells, expand it and seed it on a suitable polymeric scaffold, what you see here is a nanofiber. On top of the nanofibers, you have the cells seeded on them. Now the cells will proliferate and grow, and once you get a tissue chunk, this tissue chunk can be used to replace the damaged skin which the patient has. Okay, so that is the whole point of tissue engineering. It looks pretty simple, but there are a lot of...
Scaffolding will have any shapes, sir? What are the shapes we require?
Right. Each tissue has a favorable scaffolding feature or physical structure which is required. For instance, if you're talking about bones, bones, although they look solid from the outside, they are very porous from the inside. So, and unlike that, if you are talking about mimicking a skin tissue, so for that you need a scaffold which is more porous, both on the surface as well as inside. So a very good example for a scaffold which is used for skin graft is nanofibers. Nanofibers are basically fibers which has a diameter in the range of nanometer region. So they are very close to the extracellular matrix that the cell lives in vivo condition. Our cells in the body, they are all entangled with each other with a mesh sort of thing. And this mesh sort of thing is called extracellular matrix. Okay. So based on whatever kind of tissue which you are handling, you have to study what is the composition of the extracellular matrix around these cells. And when you are designing a scaffold, you have to come up with a composition which mimics that condition. And for each tissue, it's different. So it all depends on the physical characteristics of the target tissue which you are trying to mimic in the scaffold. So that will change. And when you change the physical property of the scaffold, which you want ultimately, obviously the fabrication techniques and all those things will be entirely different for each case. Okay, so moving ahead further. So although the cycle of events looks pretty simple and straightforward, there are a lot of complications in this process. First of all, you have to know what kind of cells you have to collect from the patients, and what is the composition of the growth culture, growth medium, which you have to use to expand these cells, because each cell has a different growth media requirements. Okay, so you have to identify based on what kind of cells you have selected, you have to know what kind of growth media you have to make sure they expand well or proliferate well in the in vitro conditions.
Sir, is it cytokines are also used for growing the cells?
Cytokines are also part, cytokines are involved, especially when you are talking about differentiation or de-differentiation of stem cells and all those things. Cytokines do play a minor role as compared to growth factors. Growth factors are the major components which determine the fate of the cells. Based on what kind of growth factors you give to the cells, they can either differentiate into a different kind of cells or just proliferate into the same kind of cells. Okay, but cytokines do play a role. Cytokines have an important role to play in the immune system. Cytokines have can modulate how your immune system reacts to the scaffold or the graft which you have implanted in. Okay, so what I was telling you is the process looks pretty simple, but there are a lot of complications in designing the kind of cells, the kind of growth factor which you need for these cells, and also we have to decide what kind of polymeric material or a metallic component would you use for preparing the scaffold, and all those things, as well as finally the growth factors already mentioned. Based, if you have fibroblast, then you need fibroblast growth factors. So each cell type has a different class of growth factors. So we have to identify all those things. So just to make sure the cell has the same characteristic as it is in your body. Because if the characteristic changes and you continue to grow this and then implant in your body, it will not work out. So when you are culturing the cells in the lab, you have to make sure that it has the same conditions as it is in your body, so that it maintains its physical characteristics. Then only it can act as a suitable graft at the end of this process. Okay. So all these factors are involved in ensuring that. So now that we have spoken a lot about tissue engineering introduction, what are the different steps involved in this, I think now roughly all of you would have come to a conclusion that there are three major components of tissue engineering. The first component is obviously the cell source, what kind of cells you would take. And the second component is the growth factors, what kind of signals you have to give to the cells to differentiate or proliferate them. And the third component, which we spoke about in tissue engineering, is the extracellular matrix or the scaffold. Okay, so these three components are usually referred to as triad of tissue engineering. It forms a triangle sort of thing. So it is called triad of tissue engineering. It has, first one is cell lines or the cell source, and the other one is signals. These signals can be growth factors or cytokines and other things. And the third component is the scaffold or extracellular matrix. Okay, which includes, you can prepare a metal scaffold or ceramic scaffold, synthetic polymers or natural polymers can be used to prepare a scaffold. Okay, so these three components are basically called the triads of tissue engineering. So let's talk about the first component, which is cells. Cells are nothing but the living part of the tissue. I should not say nothing but. They are the living part of the tissue, wherein